Let's go over to page 193, please. Okay, you already did your study guide. And I don't want to go through all those vocabulary words. So do you guys have some pretty nice, simple ways of describing all those? Are there any of them you need help with? You okay? Okay. Number 2, name the organelles that play a role in biosynthesis. So give me an organelle that plays a role in biosynthesis.
Ribosomes.
Ribosomes for sure. That should right away come to mind. Very good. What's an organelle that ribosomes are attached to?
Roughy R.
Roughy R. Very good. What's another one that's involved in biosynthesis? Think Postal Service. Golgi. Very good. And when I call it Golgi apparatus, just let it go because they can be. I've heard it Golgi complex, Golgi apparatus, and Golgi bodies. You'll hear it called a bunch of different ways. Depends on what school you're at. So don't even worry about it. Okay. What's another one?
Secretion vesicles.
Chloroplast, secretion vesicles. What's an organelle that's like involved in everything?
The
The nucleus. Okay, good. All right. Number 3. What helps the cell hold its shape if it does not have a cell wall?
Cytoskeleton.
Good. The cytoskeleton, and the other one the book gave?
ER.
The ER. The ER can help with that. Very good. Number 4. How does a plant cell fight osmosis in a hypotonic solution? Turgor pressure. Very good. The turgor pressure of the central vacuole. Okay. Number 5. A cell contains centrioles and lysosomes. Is it a plant or animal cell?
Animal.
It's an animal cell. Number 6. What organelles are involved in secretion? So give me an organelle that's involved in secretion. Golgi. Very good. What's another one?
Secretion vesicle.
Very good. And then when it gets to the edge of the cell, what's involved?
Plasma membrane.
Plasma membrane, or if it's a plant cell, you might have the plasma membrane and the
Cell wall.
cell wall. Very good. Okay. What things in the cell, not just organelles, but anything we've studied, deal with the cellular movement? So if it's moving like in a fluid capacity, what do we call it? Cytoplasmic streaming. Very good. What else would be involved in movement?
Cytoskeleton.
Very good. The cytoskeleton would. Remember I just said there were some that work like lassos?
Centrioles.
Centrioles. Very good. And then this says not just organelles, but anything we've studied that deal with cellular movement. What the book listed was the smooth ER, the rough ER, the Golgi, secretion vesicles, waste vacuoles. I don't remember a test question like that, so don't worry about it. Number 8, what are the 11 major tasks that the cell must perform? We actually already ran through that, didn't we? So you need to understand what absorption is, digestion, respiration, excretion, egestion, biosynthesis, secretion, movement, irritability, homeostasis, and reproduction. What is the plasma membrane made of?
Phospholipids.
Phospholipids. There are a few other things, but phospholipids, what else?
Proteins
And cholesterol. Very good. What's the difference between a phospholipid and a regular lipid? And Zachary is not allowed to answer because he already answered this question once. What's the difference between a phospholipid and a regular lipid?
Phospholipid has the 2 sides.
Very good. The phospholipid has a hydrophilic end and a hydrophobic end. Now, you could answer it that way, or you could say, oh, it broke off one fatty acid and has a phosphate group, but that's not the important part, is it? What you said is the important part. The important part is it has one end that likes water and one end that doesn't. And that gives it that little bipolar thing going on so that it acts like a little magnet, right? And then we can have the self-reassembling plasma membrane, which is very, very important. Thank you. That was good. Let's see. What makes it possible for the plasma membrane to self-reassemble? They're looking for one end is hydrophilic and the other end is hydrophobic of the phospholipid. Number 12, a cell begins running low on food and its energy output it decreases by 20%. What kind of plasma membrane transport is affected? Active transport. Turn to page 194, please. Notice that you have to know to identify the structures in the plasma membrane. It is on your test. Can't make that any clearer. So A would be what? Phospholipid. B is what?
Protein.
It's a protein. And in your book, it actually tells you that it's an active transport site. How about C?
Glycoprotein.
Glycoprotein. How about D?
Carbohydrate.
Carb. That's what makes it a glycoprotein, right? How about E?
Cholesterol.
Cholesterol. And F?
Filaments.
Elements of the cytoskeleton.
Yeah, so you could have said cytoskeleton there, you could have answered microfilaments. There's a few different ways you could go there that would be acceptable. And how about G?
Glycolipid.
Okay, that's a glycolipid because you have a carbohydrate attached to the phospholipid, don't you? Okay? Thank you. Number 14, if a cell dies by implosion, that means it shrivels to die. Okay, so we're shriveling it up. What kind of solution was it in? Hypertonic, because the water went from more pure in the cell to less pure outside the cell, and so it shriveled up. And I would draw a little picture there personally. If I were taking this test, I'd be drawing my little picture with my arrows to figure it out. I'm just telling you. Number 15, list the 4 stages of aerobic cellular respiration in order in which they occur. In addition, note the net number of ATPs that are made in each cell, each step. So somebody tell me, what's the first step? Glycolysis. How many ATPs net?
2.
2. Okay. What's the second step?
Formation of acetyl coenzyme.
Good. Formation of acetyl coenzyme A. Are there any ATPs made there?
No.
No. What's the third step? Krebs cycle. And how many ATPs made there?
2.
2. And then what's the fourth step?
Electron transport system.
Good. The electron transport system. How many ATPs made there?
32.
Snoopy dance, 32. Woo, woo. Okay, that's the big one, right? All right. Number 16. What is the purpose of ATP in the cell? Batteries. I like it.
Energy.
It's energy, which is why we use batteries, isn't it? So very good. Just remember that. Do you know what's funny? I've been teaching that way for years, and when I looked for pictures on the internet, and I've never used pictures before in the classes, I just want you to know that. That's this year's edition. When I looked for pictures, I saw that they used charged battery and an uncharged battery, and I thought, see, I'm not the only crazy person, because it's really a good picture for what it is. It really is that we can relate to, you know? So anyway, just thought that was cool. Okay, let's see, 17. If the cell has no oxygen, what stages of aerobic cellular respiration can still run?
1.
Glycolysis. And how many ATPs can it make from a molecule of glucose? 2. Because glycolysis, it only makes 2. 18. A scientist determines a means to extract all the ADP from a cell. Why will this kill the cell?
Because no ATP can be made.
Exactly. You just took away all the uncharged batteries, so there's no uncharged batteries to be charged. Therefore, the cell's going to die. And please be clear, this is why we die when we stop having oxygen, okay? Is because our cells no longer are able to go through the process of aerobic cellular respiration, and therefore we die on a cellular level from a lack of available ATPs. That's what kills you if you run out of oxygen, just so you know. Okay, let's see. Number 19. What organelle is responsible for breaking polysaccharides into monosaccharides?
Lysosomes.
Lysosomes. Okay.